Okay, so I like to think of defense against pathogen to happen in 3 stages, and it can easily be confused with primary and secondary response, so we should not do that. So that's what we'll just do that, okay? So I'm just going to summarize so that it all kind of makes sense, and then I might discuss in detail individual parts. The first line of defense is what we can call kind of outer defenses. So that's not a specific term, but these defenses are all non-specific. Tell me if I'm going off the screen here. Okay, non-specific, and by that what we mean is that the body is just kind of defending itself against anything that happens to be non-self. It is not directed towards a particular pathogen. So not specifically HIV, not specifically against flu, not specifically against TB. But it can do that, and that's what we call the specific immune system, immune response. So that's its own category. Specific response. Making sense? Okay. So what are these outer defenses? So the outer defenses are your skin. All right. So it forms not only a physical barrier, because what you're defending against is pathogens from the environment infecting the cells and tissues of the body inside. That's what you're trying to avoid. So the skin forms a physical barrier to the entry of those pathogens. Okay, but it's also got the composition of chemicals that are released onto the skin makes it inhospitable. So you might have kind of fatty acids and things like that make it kind of acidic for pathogens, and it might be too acidic for them to proliferate. You've also got this. You've also got skin flora. So these are bacteria who, just naturally reside on the skin. Okay, so the skin is their kind of habitat, and the skin flora then basically outcompete, because the skin flora are adapted to survive on the skin. So the skin flora outcompete most, pathogenic microorganisms. Yeah? Okay, and it's a similar story in the gut. So, when we eat food, it's another way that pathogens can come into the body, and we have to realize that the gut, our intestines, is essentially part of one of the outer surfaces of the body, even though we don't think of it as such. Yeah? So
is it the large intestines or the small intestines?
Wait a minute, don't want to get this wrong. Does that make sense? Mm-hmm. Yeah? So if we think of this as the, how do we spell it? Esophagus right there, that's the entry into the digestive system. You can see how it's continuous with the outside of the body. So in many ways, it's another entry point for pathogens. Okay? But what we're saying is that the gut provides a very inhospitable environment for pathogens that are entering the body in this way. Yeah.
What do you mean gut, like the small intestines? Or
yeah. So the esophagus is going to continue into the stomach, and then the stomach's going to be continuing into the intestines, and then out through the anus. But we have to think of it as like this is another external surface of the body which is being bombarded with pathogens, and the body has to defend against that. So similar to the skin flora, we have gut flora, and we have our digestive enzymes, which can break down the proteins and carbohydrates and lipids, if they may be so, of the pathogens. Yep, and we've got very low pHs, maybe too low a pH for these pathogenic organisms to survive. Yep. Yep. We've also got mucus membranes, and we discussed this last year. So we have kind of epithelial cells, right, that they produce mucus, which traps pathogens, and they have cilia. And they have cilia, these structures that then move the mucus up through into the kind of pharynx, and then we swallow it, and then all those trapped pathogens get digested by our stomach acids and digestive enzymes and what we discussed about the digestive system. Yeah? Finally, no, that's it. Okay, so these are all the kind of, or just some of the things that will prevent the entry of pathogens into the body, but some of them might get through. Okay, so some pathogens may find a way to get through, and in that case, what are the responses that could be made at still like a non-specific level? Okay. So it's still non-specific. So we're always thinking specific versus non-specific. So if anything does get through, like say we get a cut on the skin and pathogen is able to kind of get through the physical barrier and doesn't have to compete with the skin flora, then it's in. Now what would happen next? So what are those mechanisms? Well, we have the first thing, which is an inflammatory response, okay? An inflammatory response. So if you have some pathogens, then what will happen is that they might cause some kind of localized infection of cells. The damaged or the infected cells will release histamine, and the histamine will then cause the blood vessels or capillaries, cause the capillaries to dilate, and allows macrophages to enter, so come out. So macrophages are moving around in the blood, but when the histamine causes the capillaries to dilate, it allows the macrophages to exit through the gaps in the capillary wall and enter the kind of inflamed or infected area. Okay, and here what they do is carry out the process, and this is our next kind of defense mechanism, carry out the process of phagocytosis. So any pathogen that comes in might cause some local damage, but then the damage causes histamine to be released, which causes macrophage to enter the area, and the macrophages carry out phagocytosis. Now, what is the process of phagocytosis? Kind of similar to endocytosis, but with a few kind of twists. So if this is a pathogen, a macrophage would be a quite large cell in response, and what it does is it starts to wrap its outer cell surface membrane around the pathogen. Yeah? And then eventually it will fully engulf the pathogen, and once that pathogen is inside, the organelles, which we call lysosomes, go towards this compartment and fuse with it. And when they join with that compartment, all these hydrolytic enzymes can then attack the pathogen and basically break the proteins down, break the carbohydrates down, break the lipids down, and essentially just deconstruct that pathogen into fragments. Okay, making sense? Right, so that's phagocytosis. Now, other things that also constitute non-specific responses are the production of lysozyme. So lysozyme is basically a digestive enzyme or hydrolytic enzyme. Okay, and this breaks down the cell walls of bacteria. Yeah. And last but not least, and we might run out of space here, we have interferon. Okay, interferon. And when one cell is infected, so say a virus, and this is specifically like a virus, antivirus action. Let's say a virus infects a cell. Now obviously this cell is infected and it's probably going to be eventually lysed or killed by the presence of the virus. But what it does is the presence of the virus does something or causes a change in the gene expression in the nucleus. It activates certain genes that cause the production of a protein called interferon. Okay, so that protein, the gene is transcribed, the mRNA is translated, and this protein interferon is produced and released from the cell. And when neighboring cells recognize the interferon, they then undergo their own gene expression change to produce antiviral proteins. So this cell, when it binds to interferon, signals go to the nucleus to start producing the mRNA for antiviral proteins. Yeah? And these antiviral proteins, then even if this cell does get infected by a virus, the virus can't replicate because of the presence of the antiviral proteins in that cell. Okay? Any questions on that?
If that happens, how come in HIV it still does replicate?
So, good question. Yeah, and this is going out to the public. So if you can ask easier questions, no, just kidding. So yeah, look, different viruses have evolved, and one of the ways that they can evolve is at the infection stage they have evasion mechanisms that allows them, maybe when HIV infects the T helper cell, it does something to prevent the production of interferon by the T helper cell. Okay? Or because the HIV virus is essentially covered with T helper cell membrane, the cell may not even recognize that it's been infected. Yeah? So those are all non-specific things. They don't care what the pathogen is, they're just acting against the pathogen. So now we move on to specific response, which is a specific response, immune response directed towards a particular type of pathogen. So, I mean, what we're saying is that if some of this gets, so we're saying that, you know, many pathogens bombard our body from the outside, trying to take advantage of the conditions within our body as a way to proliferate. But our outer defenses prevent most of those getting through, while some may manage to get through. Of those that do get through, they are dealt with in a still non-specific way within the body by these mechanisms, but still one thing may get through, and that is when you need your specific immune response. Okay? Because once this thing is infecting cells and is allowed to, or has an opportunity to replicate itself and cause further infection, then it may start to cause organ failure, leading to death. So we must remove the pathogen from this situation. Okay? This is where I take a break.
Break.
Cut. Action. Right, so what we are saying is that if a pathogen then does manage to evade outer defenses, internal, non-specific responses, defenses, then it falls to the specific immune response to deal with the pathogen. Okay? Now, the overall picture that we have to have in our mind is if there is a pathogen, then it can be in two places, and the specific immune response needs to deal with both of those locations. So if you have a group of cells, okay, the first place that the pathogen can be causing infection is obviously inside the cells, okay? So we have to deal with, the immune system has to be, the specific immune response must be able to deal with infected cells. Because remember, the infected cells are what the pathogen is using to replicate itself and then be able to cause further infection. And if we get too many cells infected, it's going to affect the tissues. If we get too many tissues infected, it's going to affect organs. And if organs are failing, then that means that the organism is going to fail or die. Okay? So we have to deal with infected cells and any pathogen that's outside, what I like to call free pathogen. We must eradicate it before it can get into a cell and infect it. So any response has to be able to deal with those two situations. Okay? And what we are saying is that the antigen or the pathogen is what results in the activation of, so let's just get this overall. I'm going to go into the detail of how this works, but it's the pathogen and kind of its surface antigens is what causes the activation of T helper cells. So these are lymphocytes in the body, specific type of blood cell, okay, called lymphocytes. And these T lymphocytes or T cells have a specific subpopulation of them that are called T helper cells. Now the pathogen specifically activates a T helper cell via the T helper cell having a receptor that is complementary to that antigen. Okay? So this is an antigen of the pathogen. So the pathogen has got structures on its surface, and each of those little structures can act as what we call an antigen. And T helper cells have antigen receptors, and when you activate a T helper cell, there's two things in general that it does. And we're going to go later into the detail of this, but basically an activated T helper cell results in the production of T killer cells, and the T killer cells kill infected cells. Okay, so one branch of this immune response is to generate cells that will kill any infected cells. So those cells can no longer be factories for the production of new pathogen and cause further infection. The other thing that the T helper cell does is activate B cells. And in general, summarizing here, but in general, the B cells produce antibodies, and it is antibodies that are crucial in the destruction of free pathogen. Yeah? So this is the immune response. So if everything happens as according to the plan, the T helper cells activate T killer or result in the production of T killer cells which kill infected cells. The T helper cells also activate B cells that make antibodies that cause the free pathogen to be destroyed. Infection removed. Okay? But there's also another thing that happens. The activation of the B cells and the T helper cells produces T memory cells and B memory cells. Now this is like a long-term precaution. So basically the T killer cells and the B cells were like let's take care of the problem as it stands right now, but the process also invests in the production of long-term cells called T memory cells and B memory cells, which will make a faster and bigger response upon reinfection. So if by chance the same pathogen happens to enter the body again, the T memory cells and B memory cells are there to ensure a faster and larger response to that infection. And this is so effective that you wouldn't even feel that you had been infected. You would feel no symptoms because the response is so fast and so large. Yeah? And essentially this is what we call immunity. So this is what we refer to as a primary response. The pathogen activating the T helper cells, the T killer cells, B cells, antibodies, that's all part of the primary response. But if the T memory cells and B memory cells are, and maybe I kind of over summarized here, but essentially if the pathogen comes again, then the pathogen causes production of new T killer cells and new B cells that will go on to make antibodies, and that's how the infection is dealt with again. But what we're saying is the second time this happens, and this is what we call the secondary response, this time it's faster and larger response. So more T killer cells produced, more antibodies produced, and you hardly even feel the infection. Yeah. What's it? Is it warning me about battery? Yeah, it's still recording though. Yeah, and this is a summary of the response. quite happy with that, actually.